Review 1, Gene Bank Submission 22, and is a book and is a good number of book chapter also is contribute. Actually, it is our privilege and the pleasure to get such a renowned scientist in it, and you can listen something from today's topic about it. Today, differential expression biomarker next generation sequencing data. On this, Soumita said will speak. Dr. Chinmay Panda,
yes.
Please introduce the subject and speaker also in short. What is you think the student can be helpful?
Okay, thank you, Dr. Mukherjee, for your invitation for this type of academic session. Though it is online, because it is quite informative, no doubt. And today, because this next generation sequencing, it is quite changing time to time. It is changing the system, and in your resolution and accuracy is gradually increasing. For this is the main point, and the data bank what we have received in the, and then for analysis of the data, it is quite challenging. So how to analyze the data? Huge data bank has been generated by the high resolution sequencing, and then in this way the computer science and your information bioinformatics is a huge role. His role is an important role in this type of data bank analysis, and for the initially for the analysis of the your soft copy data, and then followed by this. This is applicable. How to apply to our translation research, particularly different diseases and different diseases, how it can be utilized, and particularly by the cancer biology or some different rare diseases and some infectious diseases, not all. And during this pandemic situation, SARS-CoV-2 infection also, it's got immense importance for this type of data analysis, and in this field, computer science, bioinformatics has an important role. And so, before wasting any time, today is a quite right topic for this type of discussion during this type of our pandemic situation. And today the talk will be delivered by Ms. Soumita Seth, and today's topic is the differential expression and biomarker discovery for next generation sequencing data. And then she's the PhD student in the Department of Computer Science and Engineering in the Alia University, and she is doing her PhD on Dr. Tapas Bhadro, and I think, and also she has published a couple of papers in different peer-reviewed journals, and then that means he has quite wide experience in this field, and I hope that from this type of deliberations we can learn a lot of things from her, how her work is progressing and what she is thinking in this line, and how to do this type of her analysis, particularly the translation research for the benefit of the mankind. So, Ms. Soumita, this is yours. Please go on for deliver the lectures. Soumita, please.
Hello.
Yes, Soumita, please.
Hello. But
yes, you are audible.
I'm audible, but my video is also on. But I don't know. Is it now? Now I am visible, I think. Am I visible?
Yes.
Okay.
Yes.
Good evening, everyone. Thank you, Dr. Dattatreya Mukherjee, for inviting me in this talk. Thanks a lot. Also, I am thanks to my respected Professor Dr. Kollan Mukherjee, Dr. Chinmoy Kumar Pandas for introducing me in this field, and also thanks to my supervisors, Dr. Taposh Bhadda and Dr. Saurabh Mollik, for whom I can come here and get such opportunity to deliver my talk, my work in front of you. And last but not least, thanks to my beloved peoples who are joining in this session. Hope I will help you by my work so that you can also get interest to work in this field in future. Okay, so can I start my presentation? Can I start my presentation?
Yes, go ahead.
Okay, thank you. I am starting.
Yes, go ahead.
Go ahead. Please unmute other persons from back. The sound is coming up. Please unmute. Mute. Miss Somita said.
Yes,
Hello?
sir, I am sharing my screen.
Yes, start sharing.
Yes.
Yes. Full screen. Give full screen. Yes. Full screen. Okay. Now it's started.
Okay. So I am
Okay, start.
Yes, I am Soumita Shet, PhD research scholar from Alia University, Kolkata. Today my topic is differential expression analysis and biomarker discovery for next generation sequencing data. This what is the next generation sequencing data? I will discuss later, but first I am discussing what is the differential expression analysis. This is next generation sequencing data is nowadays the analysis of next generation sequencing data is too much increasing. From 2021 to 2028, the researchers are very much interested in this next generation sequencing data in their day-to-day analysis. What is differential expression analysis? This is basically the identification of genes. The genes are different type of genes, like they are significantly different quantities from a particular group or a group of samples. There is some biological interference also, like that the genes may be the drug treated versus control. That means some genes are drug treated and some are the control. Then disease versus healthy individual. Suppose there is two types of the gene. One is the normal gene, one is the COVID affected genes. Different types of tissues, different stages of development and something else. Those have the significantly different are called the differential gene. And from this differential, there is some methods to analysis the RNA sequential data. Using this method, we can find out the differential gene expression. These methods are parametric method and non-parametric. Some methods are parametric method and some are the non-parametric method. Parametric method means they follow a particular distribution. They capture all information about the data and then within the parameter they follow a particular distribution. But in case of non-parametric model, they consider that data distribution cannot be defined from a finite set of parameters. So they can increase in volume at anytime. There is no particular distribution. Suppose here the some are the non-differential expression, that means they are not the different, they are normal expression, and some up-regulated expression, some down-regulated expression. Suppose there is a label, suppose 0.05, this is the label, and up the label, this is called up-regulated gene or up-regulated expression, and below the label, this is called down-regulated gene. Now, in this picture, red are the up-regulated gene and blue are the down-regulated gene, and the black is the non-differentially expressed gene. So there is several software tools we can use for differential expression analysis. Suppose DESQ, this is a software tool. This is the R tool or bioconductor package is available in online. Anyone can use who know the R software can use those software tool like HR, Lima. Lima is a very important software tool. Widely used software tools. Apart from this, there is many software tools for differential expression analysis. What is Lima? Lima stands for linear models for microarrays analysis. It can use RNA sequential, but before using Lima, first we need to normalize our data. There are several type of normalization like log normalization, boom normalization. So any kind of normalization we can use before applying Lima. And essentially, the expression of each gene is modeled within a linear relation. How we can express a model in a linear expression? Suppose this is a linear model, so it has some dependent variable and explanatory variable or covariable. So now Y is our observed data and X is our design matrix, beta is our vector of parameters to estimate, and epsilon is the random error. So our linear model expression is like that: Y equal X beta plus epsilon. Next, our topic was biomarker discovery. What is biomarker discovery? Biomarker discovery, basically, what is biomarker? Biomarker is nothing but the differential express gene, which can identify the disease gene, or which can identify the gene that are significantly different from other. Biomarker discovery research encompasses widespread in different domains, including biomedical domain. Gene marker discovery has a huge impact to detect critical disease like cancer. Using the cancer biomarker, we can do so many work like response to treatment, screening, different diagnosis, monitoring progression of disease, risk assessment, prognosis, therapeutic targets. So many work we can do using the cancer biomarker. Cancer biomarker means the significant gene of cancer, significant differentially expressed gene of cancer. Which we can use as to find out the patient is a cancer patient, whether the patient is cancer patient or not, whether the gene is cancerous gene or normal gene. And there is so many machine learning approach which is available to identify such type of genetic marker. Now, this is our increasing trends of research in next generation sequencing data analysis. From 2019 to 2026, it's showing there in various countries like North America, Europe, Asia, Middle East, Africa, South America, all. In present situation, researchers are very much interested in this next generation sequencing data analysis. there is those are the ongoing research in this domain. Especially next generation sequencing data, including RNA sequencing data, bulk RNA, single cell RNA. What is difference between bulk RNA and single cell RNA? I will discuss in my later slides. Single cell RNA sequencing technology are used to enable the large scale whole transcriptomic profiles to each individual single cell in a cell population. The interesting computational research on single cell RNA sequencing data are rare cell detection, parse matrix dimension reduction, differential approaches on bioinformatics based on statistical tests like t-test, lemma test, ANOVA test. These are the opening research area for the future researcher. What we have developed so far, we have analyzed the next generation sequencing data like DNA methylation data, single cell RNA sequencing data. This is the platform. These are the several kind of next generation sequencing data like RNA sequencing data, DNA sequencing data, targeted DNA sequencing data, ATA-AC sequencing data, link RNA sequencing data, microRNA sequencing data, single cell RNA sequencing data, ribosomal sequencing data. So there are so many types of next generation data are available to work, to analyze. This is the basic workflow for the beginner who are the new in this field. For them, the actual following workflow will be like that. First, we have to collect the biological samples or library preparation. Also, there is some weighted lab. Using from this weighted lab, we normally get the data, and need to collect this data. Then we will read all the sequence reads, and then need the mapping or quantifying. So there is some process of mapping, normalization is there, or there is some mapping like find outing the highly variable features, find the features, various type of feature selection methods are there. So different kind of mapping procedure or different type of regression procedure we can use in this step. And after that, we need to find out the DGE, differential gene expression. Normally we use a art tool for differential gene expression. Rather than R, we can also use Python for differential gene expression. And after that, we use some functional analysis on this data like classification, clustering. There are some pathway analysis, half gene network analysis, all kind of functional analysis is there, which we can do on differential gene expression. Today I will discuss my two work which I have done in my this PhD session. Last two work I have discussed here. Firstly, I am discussed this work, which is the developed a framework for detecting reliable genetic alteration in cancer using DNA methylation and gene expression data based on linear regression and deep learning approach. This is the reference. That means this paper I have published in this journal, Gene Journal, and this is the also a Scopus Index journal. I have published my paper in this journal. So now I am discussing the framework which I have developed in this paper. What is DNA methylation? DNA methylation is a promising biomarker in cancer detection and cancer classification. Two kinds of analysis are available for DNA methylation: single differentially methylated gene finding and differentially methylated region finding. But there is some shortcomings in this DNA methylation. So there is two to work as multifunctionalities from different direction, we need to incorporate different factors to correctly interpret DNA methylation data, so that it can work in multifunctionalities from different directions, such as prediction of gene expression using DNA methylation, differential expression analysis, cancer classification, half gene finding, and among others type of work. The shortcomings of DNA methylation data of the already existing method was not easy to determine directionally of the evaluated gene expression estimated from DNA methylation. An accurate measure of gene promoter methylation is difficult due to the variance in size. The size are not the same. There is too much variance in the size, as well as the presence of distal augments, like this initiates some biasness in the association of methylated region and gene model. There is also a high probability of selecting gene due to the nearby differentially methylated CpG sites and overlapping with other gene. Some specific tools are there like GRADE, Galaxy, etc., but it's create more complication in usage. Here in this work we used our disease name was the cervical cancer. What is cervical cancer? This is a type of cancer which is basically a woman cancer. Starts in the cervix, a hollow cylinder that connects the lower part of the uterus to a woman's vagina. Most of the cervical cancer grow in the cell of the outer surface of the cervix. The normal symptoms of cervical cancer is very normal, with the which is normally occur in the menstruation time or after having sex, and it is nothing but the abnormal bleeding, pain in pelvis, as well as pain during the urination. This is our workflow we have followed to develop this framework. First, we have taken our data. This is our data for cervical cancer methylation dataset, NCBI accession number GSC 30760. Our dataset have 27,578 genes, and this dataset includes 63 uterine cervical tumor and 152 normal samples. 63 was the uterine cervical tumor samples and 152 was the normal samples. And first we have taken this data. Then our next step is the pre-processing step. First, we eliminated the CpG sites that have missing value in more than half of the samples. Then we applied our new traditional quality control R package in mix, which widely useful in human methylation data analysis. Then our method we used our method to discuss discard the outliers or noise and to replace missing values using the popular k nearest neighbors algorithm. Preprocessing DNA methylation data through eliminating the outliers and replace the miss value by KNN. Then we computing, next step was computing predict expression scores of gene through the linear regression analysis. Utilize the linear regression algorithm to measure the impact between DNA methylation and gene expression of uterine cervical cancer on process DNA methylation and corresponding TCGA CESC cancer type. There is here we have two types of data: DNA methylation data and TCGA CESC cancer type data. And then we have generated the predicted gene expression matrix for provided genes and CpG site using DNA methylation data. We have applied here the tenfold cross validation to validate our model through checking the quality of gene expression inferred by linear regression model. Here we have used linear regression, and our independent variable is DNA methylation data, and the dependent variable is our TCGS cancer data. For conducting downstream validation, we compared the gene expression with the RNA sequential data. Next, we have used the Boom normalization and identifying differential express gene using lemma. Here we use the tool lemma. Here we use the regression lemma, statistical tool lemma for our two sample which are considered here control sample and cancerous sample. Lemma software was based on the supervised learning model empirical based test. Use lemma to identify differential express gene between disease and control groups. The performance of lemma is good in normal distribution as well as non-normal distribution for any sample. That's why we have chosen lemma tool here. Using the moderated t-statistics of lemma, corresponding false discovery rate adjusted p-value have been computed. What is the p-value? Probability value, which normally the low probability value, that means the genes is highly differentially expressed, and cumulative distribution function CDF and t-table. If FDR less than 0.001, that means the FDR means false discovery rate, which is less than 0.001, that means the gene is termed as differentially expressed gene. All the differentially expressed gene were considered as a single potential gene signature and could be verified at classification analysis through deep learning. Here we have used deep learning classification on differential expressed gene. This is our after preprocessing, this is our TCGA cancer type data, and we have applied the linear regression here and got the predicted gene expression data, and after that we have applied Boom normalization and got normalized predicted gene expression data. Then we applied here empirical based test using lemma where FDR less than equal 0.001, and we find out the differentially expressed gene 6287 differentially expressed gene we have got. Now on this differentially expressed gene we have applied deep learning on training set, and first we have divided it training set and test set, and we have used 10-fold cross validation on all samples, and we applied deep learning analysis. We have used here NNet. Deep learning NNet, which is the feed-forward neural network-based model, and we with 10-fold cross-validation to examine the class-level normal and uterine cervical cancer group. There is two group, normal group and uterine cervical cancer group of differentially expressed gene, and we repeat our learning procedure 30 times and ran NNet tool using a certain number of epochs, which is termed here max iteration, which is nothing but the epoch value, and deep learning methods was internally repeated for this maxed time. Here we have taken 10 as a epoch value, and then we have taken 100. 30 times is repeated, it is taken 100 as epoch value. It is your choice. You can choose as in your choice how many iteration you will choose. It's completely in your choice, and then you computed the classification matrix at one time iteration for each fold. Obtained a confusion matrix. What is confusion matrix? Which have the four indices. One is true positive, false negative, false positive, for true negative. True positive means the diseases are already. True positive means the normal training set and the test set both are the same. That means suppose the patient is COVID positive and the report is also the COVID positive. This is the case of true positive case. Similarly, false negative means the patient is COVID positive but falsely the report is negative. So this is the false negative case. And similarly, false positive means the patient is negative, COVID negative, but the report is positive. Then this is the false positive. So actually, we want we show for better result it would be true positive and true negative. The true negative means the patient is also negative and the report is also negative. This is the true negative case. And we have repeated for each fold of samples. That means we will take one and as it is the tenfold cross validation, so we will take one and there is other 9-fold sample. In this way, this will be repeated 10 times and added all these matrices for this 10 times internal repetition and then proceed a final confusion matrix. Repeated the entire process the multiple times, 30 times to obtain the average classification matrix value like average accuracy, average sensitivity, average specificity, average precision, average overall error rate and area under curve. Next in the flow chart, after we already applied the deep learning for classification.
Hello ma'am. There is a question. Can you please explain the false positive once again, please?
False positive. False positive means
Yes.
suppose you know there is a patient who are not just they have some fever. He has some fever. So he thought that he may be the COVID positive patient, but actually he is not the COVID positive patient. But in his report, this is showing that this is the positive. Wrong report. This is the false positive case. Understood? Hello?
Yes, ma'am.
Yes, ma'am.
Thank
That
you.
means the report is wrong. The report is wrong. The patient is not the COVID positive patient. He has some symptoms and the report is wrongly telling that the patient is positive. But actually he is not the COVID positive patient. This is the false positive case. And true positive means the patient is also, the report is positive and also the patient is COVID positive. This is the true positive case.
Excuse me, ma'am.
Tell.
Ma'am, without the presence of virus, how is it detecting positive in report?
Please repeat.
Ma'am, in case of false positive, without the presence of virus in the human body, how is the report showing that virus is present and is showing positive in report?
This is the wrong. Sometimes the report may be the wrong. This is a research process, so sometimes it happens. The report may be the wrong. So that means
Excuse me, I am highlighting something. One point: false positive, true positive, false negative. It's a test wise. Now, what we are telling. 1 minute, I am clarifying this. This is a patient is positive, report is negative, patient is a false negative, and report is positive, patient is negative, is false positive. This is a true false thing. But the question is what he has said that, how can I know that this is false? When patient attendant, the patient has got the report, and the report is positive. Is it false positive or true? But in research purpose, what we are telling, how many patients of this method, suppose the method you are doing the two methods comparison, is on a rat method and other is artificial method. If a rat method, false positive rate, false negative and false positive, that rate is not corroborated to the true positive or true negative. False negative rate is quite high. That's why rat is not permissible to all the patients. But RT-PCR patients, 99% patients are true positive cases. That's why the test, it is, in other words, in our sense, we are telling sensitivity and specificity. The tests were highly sensitive, less specific, the test is, we are using. More specific, less sensitive, we should not do the test. Most probably I can explain better this.
Yes, sir. Thank you.
Thank you, sir. So after classification, we have applied another. We have applied correlation on there to correlation on our differentially expressed gene or our biomarker to find out the hub gene network. Pearson correlation we have used to find out the hub gene network, and for network analysis we use a software tool which is Cytoscape. And here we have given this correlation value in this range 0.8 positive 0.8 to less than equal minus 0.8. These edges we will select to draw our hub gene network. This is the hub gene finding. We have applied Pearson correlation on differentially expressed gene identified by our method for finding out the active edges among genes having correlation value 0.8 and minus 0.8. Perform degree centrality analysis through Cytoscape, which is the online tool, and here we have used the score, in degree score and out degree score of our differentially expressed gene, and we have marked the top 10 in degree hub gene DGs differentially expressed gene and 10 out degree differentially expressed gene. Those are the gene name. And another one functional analysis we have done here, which is the gene set enrichment analysis. Normally we have we do this enrichment analysis for biological significance. To assess the biological significance of data and disease relevance of signature genes, we applied this gene set enrichment analysis. Here we used web-based gene set analysis toolkit software, and we do the catch pathway analysis and also ontology, gene ontology analysis like GeoBP, biological process, cellular component CC, and molecular function MF. Those analysis we have done here using web-based gene set tool analysis. This tool also, there is another tool, that is David software tool also we can use for this pathway and geoterm analysis, and we have identified catch pathways and geoterm, and here also we have give a condition of FDR less than equal 0.005. These are our differentially expressed gene, and this is the top gene in this purpose. Here the FDR value is low. In this way, we have where the FDR value is low, that means that gene is highly differentially expressed. In this way, these are the top 10 DGs. And this is our average accuracy. In this method, the average accuracy is 90.69%. Also, there is a sensitivity value is 73.97%. Specificity is 97.63%. And this is some error is there, so standard deviation value is also there, and area under cup is here 0.858. These are our all plot of accuracy, specificity, sensitivity. Now we have compared our classification method. We already used the NNet, and we have compare it with another method, that is RSNet. This is the Stuttgart neural network simulator based deep learning tool. This is also a deep learning tool, and we have applied this tool also on our data, and also we have used here tenfold cross validation, and same process we have followed, repeating the total entire process 30 times, and we got that for this existing method RSNNS, this software tool, we got 87.27% classification accuracy, where in our method we get the better classification accuracy. So our framework has better performance than all other methods using deep learning tool. This is our half-gene network structure. These are the 5 half-gene. We have measured it using in degree. The in degree is high. Using decreasing order in degree, we have taken 5 half-gene, and also similarly decreasing order in out degree, we have choice 5 out degree, 5 half-gene. These are the significant catch pathways. These are the GeoMF term, and the summary of this work is that here it's provided a framework using linear regression, differential expression and deep learning to provide correct biological interface for integrating DNA methylation and corresponding TCGA gene expression data to uterine cervical cancer. Eliminated outliers, applied linear regression to determine predicted gene expression, identified 6,287 differentially expressed gene with FDR cutoff less than 0.001 using downstream analysis through empirical based test using lemma. Applied NNet deep learning method to interpret differentially expressed gene with tenfold cross validation. Through the deep learning and comparative study, we can say that our obtained diseases are strong and efficient in disease classification. We have performed in degree and out degree half-gene network analysis using Cytoscape. Use pathway analysis on diseases with FDR 0.0 less than equal 0.01. This is the cutoff, FDR cutoff 0.001 using WebGIS. Our framework is useful for better biological interpretation of the DNA methylation data rather than single differential methylation analysis or differentially methylated region finding, because our method can do multiple work or multifunctionalities is important in our framework.
Hello, ma'am.
Hello.
Ma'am, most of our students are BSc and MSc students who are in the biology background. Can you please tell that how they can master these things, or what kind of collaborations they can make to run these type of projects or to take a part in these type of projects?
As they are biological student, so biological interference knowledge or the biological significance of this work, they are good than me. Okay, and I am completely the computer science student. So now they can do that. For collecting those data, first they can do normalization, or any kind of pre-filtering step are there. So first they can do the normalization and other phases using this data, and then they can use the any kind of software. The tools are also available in different site. If they like the art tool, then art tool, there's the bioconductor library, and there all kind of art tool they will get. So for the basic statistical part like linear regression and this type of different statistical tests like t-test, lemma test, this they can apply on this data. And as they are completely biological students, so they are know those part very well, like crossover, mutation, those part they are know very well. So using those knowledge and use some some software they can do such type of project. And also for comparison, they may use some biological significant discussion, like that suppose there is a marker, the marker is related with our gene, our disease, so this marker can relate it with other disease also, but or some other marker which is related it this disease, and some new marker, because our gene or our type of virus are evolving again and again, so some new type of gene, new type of marker they can get. This is called a novel marker. So this type of work they can do.
Thank you, ma'am. And is there any online or offline training or diploma session for mastering these softwares and all?
For if you want to learn R, so to learn R, there is many training programs there, or many online classes are there on R software. How to use R software, first how to write code in R in this type of that you can do. And another thing is, if the researcher on this field, they can help you for how to use such type of work using the software, R software in this way.
Okay, you proceed. Next. It's completed your lecture?
No.
You proceed.
Okay, now I already discussed the DNA methylation data and TCGA cancer sequencing data. Now this is the RNA sequencing data, bulk RNA sequencing data and single cell RNA sequencing data. Single cell RNA sequencing data is a very new topic. Just around 2 to 5 years, the people that know single cell RNA sequential data. So this is a completely new topic for the researcher today. So first I am discussing that bulk RNA sequencing data, people are working through 2 decades on bulk RNA sequential data. But what is different in single cell RNA sequential data? Why today people are so much interested in single cell RNA sequencing data? Bulk RNA sequencing mainly reflect the average gene expression across thousands of cells. That is in the like the picture, there is different kind of fruits, and in bulk RNA sequencing data, it's make a fruit juice, and in single cell RNA sequential data, it separate every fruit in different cell. This is the different. Single cell RNA sequential data have been applied to various species, especially to human different type of human tissues, including normal and cancer, and their studies revealed meaningful cell-to-cell gene expression variability. That means in bulk RNA data, they are make a combination of all the tissues or all the species, and then they analyze the data. But here for every cell different type data, heterogeneous gene expression, we can use the single cell RNA sequential data. For every cell we can make the expression like that. This is our tissue, and for bulk RNA analysis, first we have created a bulk RNA input, and then average gene expression from all cell. But here first we have single cell analysis. First we are giving all input, the specific cell, single cell, then for each cell there is a distinct expression profile, and then we will find out the heterogeneity and subpopulation experience variability of thousands of cells. But in bulk RNA, there is if there is heterogeneity, some problem is occurred there. We can't separate them. This is the heterogeneous tissues, bulk RNA sequencing data and single cell RNA sequencing data. First they have they extracted total RNA sequencing, total RNA sequential data, and then there is also the single cell barcode they have created, and then this is our sequencing data, and then in bulk RNA they have created a average expression data, but in case of single cell RNA they created unique transcriptome expression for different cell, diverse cell. The cells are different. Red are the completely different group, and green are the different group, yellow are the different group, and blue are the different group. And then this created a homogeneous cell population, but there is the heterogeneity and cell to cell variability is there. This is the basic difference between bulk RNA sequential and single cell RNA sequential. Single cell RNA sequential plays an important role in medical field like oncology, digestive and urinary system, and microbiology. There is the main problem to analyze single cell data is that losing cellular heterogeneity information, since it can detect heterogeneity among individual cells. Because if we create a average expression, that time there is high chance to lose the information of heterogeneity. The workflow of single cell sequencing is isolating the single cell from a group of cells. There is a group of cells. First we need to isolate all the same data or single cell data, and then we do the mapping, molecular mapping, and tracking the immune infiltration and epigenetic changes. Suppose this is our solid tissue, and from this tissue first we do the single cell isolation. Then there is some step like there are different kind of RNA type and different type of RNA, but several types of data are there, heterogeneity is there, and then they find out the all the sequencing reading, and then all the cell type need to identify it. Cell type, single cell expression profile, there is different types of cell, all cells are different, and after clustering we got the different types of cell. So here we can use clustering classification as we like, we can do here. This is my work 2, which is introduce a potential cluster specified frequent biomarker discovery framework using dimensionality reduction and hierarchical agglomerative clustering level for single cell RNA sequential data analysis. This is also published in this journal from Frontier Genetic Journal, and this paper actually not published, this is accepted. That the main challenges in single cell data are the single cell data, basically the large scale data, so they faces several analytical and technical challenges due to the high dimensionality, sparse matrix computation, and rare cell detection. The high dimensionality and sparse matrix creates a curse of dimensionality. Several technique and quality control, mapping, quantification, dimensionality reduction, clustering, finding trajectories, and identifying differentially expressed gene need to be included for the computational analysis of single cell RNA sequential data. Among them, the two techniques are very important to handle the single cell sequential data. One is dimensionality reduction and another one is clustering. This is our workflow. First, we have used single cell RNA sequential data. Here we have taken rare intentional cell type in mice. This is the NCB accession number GAC 62270. Our initial data set has 23,630 features and 1,872 samples, which are nothing but the cells. Now, in preprocessing stage, we discarded the features and cells that do not have minimum number of cells and features respectively, and we have created separate object. Why we have used separate object here? In separate object, there is also the reference in the below note. You can check here what is the separate object. Separate object basically store the data matrix, allowing us to store both data, that is nothing but the input feature sample matrix, and the analysis, which analysis we are doing here, like PCA clustering result together into a single cell data set. Together, it can store the data and the result together for a single cell data set. This is after that we use a quality control metric for cell filtration. Explore quality control based on user defined criteria for selection and filtration of cell. First we filter out all the empty cell. Then our motive was low quality, filter out low quality or dying cell. It is an important preprocessing strategy for single cell RNA sequential data. What is the low quality cell? The cells which have few genes is considered as low quality cells or dying cells. Choosing appropriate threshold to keep high quality cells without removing biologically relevant cell type is an important factor. Then we have defined a threshold range for 200 to 2500 for a number of unique feature in a cell, and filter out that cell, the cells that does not meet the criteria. That means minimum number of feature will be 200 in a cell and maximum will be 205,000. The cells which have below 200 feature, we are filtered out those. To avoid removing biological relevant cell type, we computed mitochondrial QC quality control matrix to calculate the mitochondrial count percentage from the set of feature. Also define an upper bound threshold for the percentage of mitochondrial count. Here we have take it 5% and filter out the cells above the upper bound. The cells which have more than 500 mitochondrial count data, we have taken that also and we have filtered out those cells who are above the upper bound, and upper bound threshold is 5% mitochondrial count. Next, we have used log normalization to log normalize process to data normalization. The log normalize basically divides the specific feature count of each cell by the total count of that cell and multiplies it by a scaling factor 10 to the power 4 and then performs natural log transformation. Z-score are calculated from the log normalization count. Next, we have identified highly variable feature. Which are the highly variable feature? The feature which are highly expressed in some cell and lowly expressed in other cells are noted as highly variable features. That means features are nothing but the gene. That means the gene which are highly expressed in some cells and also those are very low, absent or lowly expressed in other cells, they are the highly variable features. Highly variable genes play an important role in downstream analysis in single cell dataset through highlighting the biological signal. We use a mean variance relationship model VST to identify highly variable features. We have ranked the feature using maximum standardized value. That means the maximum variance means highly variable, and have chosen 2000 top features which are the high variance. Linear transformation and linear dimensionality reduction. First we are doing here linear dimensionality reduction method to accept high dimensional data as input and project them to an optimal low dimensional base. feature selection and feature extraction. These are the part of linear dimensionality reduction. Here we have used PCA, principal component analysis, for capture covariance as feature interest. Before applying PCA, we perform linear transformation to standardize our data. PCA technique maximize interpretability and minimize information loss simultaneously. In this way, we have identified 50 top significant principal component based on a strong enrichment of low p-value features. Next, we have used clustering. We have done clustering on cells, that is cell clustering. We have applied here graph-based clustering on our data and considered the first 10 principal components for cluster analysis. We obtained k-nearest neighbor graph by Euclidean distance in PCA space. Calculate the edge weight between the two cells through the Jacob similarity, which is defined by shared overlap between cells. And we applied modularity optimization algorithm, agglomerative clustering technique. What is agglomerative clustering? That means first we have started from first we have dated from 1 point, and then we will do the, suppose we have 10 points, so 1, 2, 3, 4 to 10, there is 10 cluster, and then we will find out the similarity and we will merge them accordingly, and at last we will get a large cluster. This is called agglomerative clustering. Clustering has two types, agglomerative and divisive. This is the process of agglomerative clustering. And what is the modularity? Modularity basically there is the high interaction within a cluster and low interaction between the cluster. It is the modularity normally measured by the interaction within a cluster and the interaction between two clusters. And if the interaction within a cluster is high, then modularity value will be high. And in modularity optimization method, our goal is to maximize the average modularity for computed cluster. Next, we have finding out cluster specific biomarker. We have applied here the model based analysis of single cell transcriptomic tool, which is the MAS. This is a statistical tool for single cell sequencing data. And this MAS tool we have used to find out the cluster specific biomarker. So first we have considered each cluster as one group. This is our experimental group, and the other clusters, all rest clusters are another group. This is our control group, and we repeated the procedure for all the cluster to find out the differentially expressed gene from each cluster. Then we have detected cluster specified biomarker based on the p-value and Bonfermi corrected adjusted p-value, which is also called false discovery rate, FDR. And our main goal here is to find out the overlapping marker. That means the marker which are present more than one cluster. It's called the overlapping cluster. And if suppose there is a 10 cluster and a marker is the present in 8 cluster, so this is the this marker is the most frequent cluster marker. In this way we have find out our frequent cluster markers. This is our result, and we have obtained 10 cluster here with the maximum modularity 0.8851. Modularity value always lie between 0 to 1, and 0.8851 is our maximum modularity for this data, and we have got the 10 cluster. And we use nonlinear dimensional reduction technique UMAP to visualize the similar cells of graph-based cluster in low dimensional space. This is our cluster specified top frequent biomarker. Here we have got for these 3 biomarker we got 5 frequency 5 by 10. That means we have 10 cluster, and this marker is present in 5 cluster. Cluster 0, and when we compare cluster 0 and cluster 2, group 1 is cluster 0, and cluster group 2 is the rest of all cluster. That means this cluster we got group cluster 0, cluster 1, cluster 3, cluster 6 and cluster 7. This 5 cluster we got this marker. In similarly for this marker also we got in 510 cluster 2, cluster 3, cluster 6, cluster 7, cluster 8. In this way we have find out the top frequent biomarker. This is our hub gene network, and also here also we have do the pathway analysis, and the advantage of our framework is that our hub gene network reported the 5 highest degree gene, which are those which are nothing but the ribosomal protein gene. In single cell analysis, ribosomal protein genes are most highly expressed genes in most cell type. So these ribosomal protein genes play an important role for cell growth and proliferation. We have tried develop effective tool for single cell dataset which can enable similarly transformative advances in our ability to analyze and interpret single cell data. These are the limitation of our framework, and there is also the future scope. Anyone can we will obviously extend our work, and anyone can do their research work from this. As a shortcoming of our framework, here we have used raw data matrix in place of imputed matrix. In our future study, we already working on a problem where we have using the imputed data matrix, and also beside this we will we want to extend our current work in here we have used single objective clustering algorithm, but in our next work we are interested to use a multi objective optimization technique in clustering procedure to obtain a better clustering result. It can also be applied on big data analysis, rare cell detection and single cell RNA sequencing data analysis, big data rare cell detection in single cell RNA sequential data analysis. This is the normal analyze procedure of next generation data. I am repeating for the beginner. If suppose you have some gene expression, and using this gene expression you can make a network, different type of network construction you can do, biological network you can do from the gene expression, and then you can identify several module using classification or clustering, and then there is several work you can do on this module, like regulatory network identification, differential co-expression analysis, finding hub gene, enrichment analysis, different type of prediction analysis, different kind of work, different kind of research work you can do on it, and then you can also do some biological comparison, like with the potential disease gene and the disease associate model, and what is the which gene is the more important for this disease, which kind of identification of identification of prime gene, identification of prime marker you can find out using this data sheet. Future scope of research in this field is signature detection, rare cell identification, sparse matrix dimension reduction. These are our reference. Thank you for listening me.
Thank you, Dr. Panda.
Yes.
Yes.
Shomita, thank you for your excellent lecture. And you have discussed a lot of things from the for the beginners to the beginners those who are in the bioinformatics line in the beginning. They can also be benefited from your excellent lecture, and also from the other researchers also. And now, is there any question? And how can we proceed? Is there any question?
Dattatreya, you proceed about the question.
Yeah. Hello. Hesika, are you there? Yeah, I can see you. So, Hesika is a very attentive participant, and she has attended all the 3 lectures very actively. Thank you, Hesika. So, her question is that, thank you, ma'am, for the insightful content. Ma'am, could you please share any tips to finding Google good journals for publishing research work based on bioinformatics, biotechnology, or biological science based on your experience?
Good journal for bioinformatic field, I will tell Nature is one of the good journal for this domain. Apart from that, you can choose a journal. IEEE transaction is always good, and there is some good open source open access journal also, like Frontier is a good open access journal, and also there is other like there is other. And for finding out through Google, I will tell to find out the first the impact factor of the journal, and search the good journal. I know there is some also bad paid journal also, but some open access journal are really good. Elsewhere is one of the good
Thank you
journal.
so Shomita ma'am. Chinmay sir, you have also lots of experience in publishing papers. So sir, the main problem we student usually face, that is that we don't have any funding. So can you please give some insights on how we can publish in a free journal like PubMed free journal?
Because nowadays is a big problem for our country, or like our country or other sources. Those funding is very poor funding. So in that case, the category related, the topic related journals you have to choose. Like for example, in case of cancer, there is one journal, International Journal of Cancer, this is published by UICC. It is a free journal, and then some other elsewhere they have some free journals, some paid journals, some free journals also, and then you have a BMC groups, they have some paid journals. So in the case elsewhere journals you can proceed, and in International Journal of Cancer, that is required UICC, that is required free journal, and then in some cases if you have